and research series, How Your Gut Health Can Affect Your Blood Cancer Treatment and Quality of Life. The community of microbes, our bacteria, fungi, viruses, and their genes living within our digestive tracts, known as the gut microbiome, can play a role in blood cancer treatment. In today's presentation, Dr. Bertrand Routy and Dr. Ariel Elkrief will speak about this exciting area of research. My name is Paul O'Connell. I am the Research Program Lead at the Leukemia and Lymphoma Society of Canada, and I will be your host for today's webcast. Since there are so many of you who are in the audience, we invite you to type your questions in the question box of the Zoom webinar panel. An LLSC staff member will monitor the questions at the end, throughout and at the end of the presentation. I will read some of your questions aloud at the end of the presentation during the question and answer period, but please keep in mind that since we have such a large audience today, we may not be able to answer your questions. Also note that Drs. Routy and Elkrief will not be answering personal health questions or questions that are not related to the topic. Today's presentation will be recorded so that you can listen to it again on our website. But before we begin our main presentation, I'd like to share our mission and highlight some resources that might be helpful for you or someone you know. At the LLSC, our mission is to cure blood cancers and improve the quality of life for those touched by a blood cancer and their families. We listen attentively and with compassion to the challenges you are facing. We can point you to the information specific to your type of blood cancer or treatment. We help you get organized to manage and take charge of your care, and we can identify services that will be beneficial to you. Do not hesitate to contact the community service lead in your area to learn more. Our website, bloodcancers.ca, was designed with you in mind to ensure easy and personalized navigation no matter your current circumstances. Next slide, please. On our website, another resource that can help you is the LLSC Health Manager app. This free app can help you track side effects, medication administration, food, and hydration. The app also helps to offer medication reminders and has a nutrition meal planner along with many recipes to help you get the nutrition you need. The app also allows access for a caregiver and is free to download in the App Store on Apple or Android devices. We have two great support programs to support the blood cancer community. The Peer Support Program is a support service that matches people affected by blood cancers, caregivers, and their families with trained volunteers who have been touched firsthand by a blood cancer and shared similar experiences. Whenever possible, participants and volunteers are matched based on their age, gender, and diagnosis, or the issue that is most concerning to you. Contact the Community Services Lead in your region to learn more. Our virtual support groups from coast to coast are professionally facilitated and offer a safe place to share experiences and to inspire and encourage others also affected by blood cancer and their caregivers. Please visit our website, bloodcancers.ca, to learn more and join. It's now time to introduce our speakers today. Dr. Bertrand Routy. Sorry? Dr. Bertrand Routy and Dr. Ariel Elkrief. Dr. Routy is a clinician-scientist and associate professor in the Department of Hemato-oncology at CHUM at the University of Montreal. Upon his recruitment to CHUM in 2018, after completing his PhD with Dr. Laurence Zitvogel, Dr. Routy quickly established himself as the scientific director of CHUM Microbiome Centre, where he began his work to develop novel microbiome-based therapeutics in oncology. His work contributed to the discovery of the gut microbiome as a novel prognostic biomarker for immune checkpoint inhibitors, or ICI, in various cancers. He characterized the deleterious impact of antibiotic-related gut dysbiosis in ICI outcomes, which led to pivotal changes in clinical oncology practice. Dr. Routy has demonstrated that modulation of the microbiome by fecal microbiota transplantation, probiotics, and prebiotic supplementation has the potential to circumvent ICI resistance. His team currently leads several microbiota-centered trials in oncology, ranging from phase 1 and phase 2 trials with the aim of decreasing primary ICI resistance. Dr. Elkrief is a medical oncologist and clinical researcher at CHUM, where she focuses on clinical studies on the microbiome. She recently received the Cancer Research Society's Next Generation of Scientists Award and the American Society of Clinical Oncology Young Investigator Award for her research. Thank you for being here today.
Thank you for having us. We're very happy to be with you this afternoon. Thank you for joining us. So our disclosures, but we work with most of the big pharma and the biotech company, but this will not change much our presentation today. So today we're going to talk about bacteria. And again, for most people, when you think of bacteria, you think of Louis Pasteur in Paris, who discovered the first bacteria. And then you think of Fleming, who was the first one to, and then received the Nobel Prize to develop antibiotic in order to kill the bacteria. Because in your mind, when you think of, oh, I have a bacteria, meaning you have an infection, could be in your lung called a pneumonia, on your skin infection, and your bladder, it's urinary tract infection. And so the field in medicine and throughout my training, for me, bacteria are equal to bad and that needed to be killed by antibiotics. However, we have forgotten, and including myself throughout my medical training, that we are covered with bacteria. And this is actually a plate where my son gave a kiss to this plate. And I just wanted to show him that he is covered with bacteria because you see his lips and all the white things are actually colonies of bacteria. And interestingly, you see that we are covered with bacteria, and the most important or the most number of bacteria are actually in your gut, where you have more bacteria than number of cells, more genes in your gut than in your entire body. And we will mostly focus on the bacteria that are present in your colon. Again, some bacteria are important in the mouth for the development of cavity, in female reproductive tract, and in the skin. There's also some evidence of the importance of these bacteria, but again, most of the talk will focus about the gut. And so how the gut microbiome change or evolutes over time. So interestingly, so as you know, when you're born, either you born through a natural way and you benefit from the vaginal microbiome of your mother. And we now understand that children that are born through C-section miss this opportunity to be faced with bacteria. And the first 2 years of your life are actually key in order to be exposed with bacteria. And what I say key is because when you're born or within the first 2 years of your life, your immune system is developing and you're getting used to see foreign particles, foreign things that do not belong to you. And there's more and more evidence that our children are too clean, and it's called the hygiene theory. And especially post-COVID, I can tell you that my now 3 sons, they wash their hands all the time, and they're obsessed with hand washing because they lived through COVID. But what we are now understanding is maybe in developed countries, because we have been so clean, the food that we eat is treated with antibiotics, we wash the food all the time, we actually see an increase of autoimmune disease such as asthma, multiple sclerosis, and Crohn's disease, which is a gastrointestinal disease. So there's a lot of literature to really show that bacteria or certain bacteria are actually good for you and for your health. And so over time, how your microbiome changes. So when you become a teenager to around the age of 65, there's not a lot of change because human adults don't like to change most of the time. We eat the same thing. And you can see that your microbiome is changing when you receive antibiotics. So most of people develop diarrhea, and this is because you have a change in your microbiome. If you go to the Caribbean for vacation, you clearly see that your microbiome changed. But interestingly, when you go back to your normal house, you eat the same thing, your microbiome goes back to its baseline. And so what's interesting is that most of your immune cells, so immune cells are there to protect you against infection, against COVID, against bacteria, but also against the development of cancer, are actually living along your GI tract. And every day, the immune cells are seeing this bacteria. And the philosophy or the hypothesis behind having good bacteria is that if you're exposed to a lot of bacteria, your immune cells are used to see foreign or not yourself bacteria or cells. And then if you have one attacked by one bacteria that is not harmful, but it's just new, your immune cells will not overreact and attack yourself, which are all the autoimmune disease, where it's your own body that attacks itself. And what about cancer development? It's probably too early to comment, but I'm sure you've seen in the press, especially for colon cancer, now a lot of young patients develop colon cancer. And of course, I have this big question mark when I see this review paper. Is it due to the diversity of the microbiome? But again, way too early to confirm this hypothesis. So over the last 10 years, the gut microbiome has rapidly shifted from a joke. And I can tell you, so I did my training at Princess Margaret in Toronto, and then I did a PhD at Gustave Roussy in Paris, which is the biggest cancer center in Europe. And you can see, so this is a biography written from the editor of Nature, where all the grand professeurs in Paris made fun of us when my mentor, Laurence Zitvogel, told me you have to collect fecal samples of patients with, at that time, melanoma and non-small cell lung cancer. And what I will walk you through with my colleague, Dr. Elkrief, is that over the last 10 years, 4 major discoveries really propelled the microbiome from a joke to one of the hallmarks of cancer. So Anand wrote this very now famous paper 2 years ago, which are the key factors, the key determinants for cancer development and response to cancer. So let's go back now to bone marrow transplant, and this talk is going to mostly focus on hematological malignancy. So I just want to remind you that the first cell transplant was performed in France, in Paris, by Professor Maté, where 5 scientists were exposed to radiation. And then they knew that their bone marrow was not going to produce any white blood cells and red blood cells. So they were shipped to Paris, and Professor Maté took stem cells of random donors. And you'll see that Dr. Elkrief is actually doing sort of the same thing right now. They're taking healthy volunteers, and they infuse these stem cells to these 5 scientists. And interestingly, if I remember well, 2 out of these 5 scientists survive. And it took actually 25 years to understand the HLA compatibility. So why certain cells from a healthy volunteer can fit you and can become your new immune system without being too deleterious, and we'll talk about that. And this led, of course, to the bone marrow transplant and allogenic stem cell transplant, which still remain the corner treatment for patients with acute myeloid leukemia and myelodysplasic syndrome. And so, as I told you, in bone marrow transplant, you receive the cell of someone else. And of course, this someone else should not be your exact twin, because if you get your exact twin cells, they have the same cells. And the advantage of taking the cell of someone else is that your body did not recognize your cancer because you developed leukemia. So if you take the cells of someone else, these new immune cells will recognize the leukemia cells and will destroy it. And that's the basis of bone marrow transplant. However, the new cells are not your cells, and they're not perfect, and they're not perfectly intelligent, and they're not able to recognize only the leukemia cells. They also recognize yourself as someone foreign because you're not HLA matched at 100%. And this leads to graft versus host. And unfortunately, despite all the treatments, all the development in the field of bone marrow or allogenic stem cell transplant, graft versus host remain the major hurdle. And most patients, unfortunately, still die from graft versus host development. So this is for allogenic stem cell transplantation and leukemia and MDS. Now you probably have heard for CAR T cell, and we'll talk briefly at the end of the importance of the microbiome in CAR T cell. But so this is for the treatment mostly of lymphoma and ALL, so acute leukemia, lymphoid leukemia. So this time we take your own T cells. The company will generate a new receptor on the T cells that will attack the CD19, so the receptor on the leukemia or lymphoma cells, and the cells will be reinfused to yourself, and they will then kill the cancer cells. So the advantage here is that it's your own cells, so there's no graft versus host disease because it's your own body that does not recognize you as foreign, but there is other types of toxicity, mostly cytokine release syndrome, meaning that all the cells are working so well that there is an inflammation and the patient can actually have a lethal complication. So what is the first demonstration in hematology that the gut microbiome is important? So this is actually an old paper from 1974, where they demonstrate in mice. So in mice, we're able to reproduce allogeneic stem cell transplantation. And in this experiment, they took normal mice and compared to germ-free mice. So germ-free mice, and Dr. Elkrief will show you a picture of her animal facility, but germ-free mice are mice that live in isolators that have never seen any bacteria in their life. And you see that if you have no absence of bacteria, the patients were actually, or the mice, sorry, were actually doing better. So this was the first demonstration that the microbiome is important in allogenic stem cell transplantation. And then the first discovery is from the team from the Memorial Sloan Kettering, where they demonstrated that the baseline diversity. So I told you initially the hygiene theory, the more bacteria you have, the better probably you'll be, or the less risk you have to develop autoimmune disease. The group from Marcel van den Brink went to another layer. They said that if you have a lot of bacteria before you get your stem cell transplant, you will actually live longer. And interestingly, you will live longer because you will not develop toxicity related to the bone marrow transplant. So of course, in medicine, we like to see a validation cohort, a lot of people, a lot of patients. So here you see there's only 26, 30, and 34 patients. So subsequently, they published a follow-up story, this time with 1,360 patients from 4 different large academic centers in different countries. And once again, they demonstrated that patients with baseline high diversity, so a lot of various, again, good bacteria, non-pathogenic bacteria, were actually living longer compared to those with not a lot of bacteria. And of course, as you can expect, for those who went through or a family member went through bone marrow transplant, once you receive your treatment, you get the chemotherapy, you get antibiotic, your diversity goes down very rapidly. And what's quite interesting in this paper is that, once again, the increase in overall survival was not due to the increase of cancer destruction by the new immune cell. It was due to the fact that you had less toxicity and therefore you were living longer because the new stem cells were not as active against you, so therefore less graft versus host. So actually, when I was a medical trainee, I was at Princess Margaret and I did an elective rotation at CHUM at Maisonneuve-Rosemont in Montreal. And I was intrigued because at Maisonneuve-Rosemont, they were giving antibiotic before allogenic stem cell transplantation. And I knew about this paper demonstrating that the higher diversity you had, the better you were doing. So we published this first paper where we demonstrated that patients that receive antibiotic develop more severe graft versus host, so grade 2, grade 4. So this means that you need treatment such as prednisone or other treatment to overcome this graft versus host. And surprisingly, once again, if they receive antibiotic before their allogenic stem cell transplantation compared to another protocol where there was no antibiotic in the protocol, patients were living longer. And remember this graph because Dr. Elkrief will show you similar data for patients treated with non-small cell lung cancer and other types of cancer. Then the group of New York went one step further than me. They said that, okay, so Bertrand showed that antibiotic before allo stem cell is not good. Us, we're going to look after because, as you may know, once you get a bone marrow transplant, an allo transplant, usually during 1 month, you are neutropenic, meaning you have no white blood cells while the new cells are generating and developing a new immune system. Therefore, most patients going through allo stem cell require antibiotic after they receive their transplant. And interestingly, again, the group from New York demonstrated that when you have an infection or when you have no white blood cell, you're called neutropenic. We, as clinicians, give broad spectrum antibiotic, meaning the largest antibiotics that are able to kill everything. And in this nice paper, they demonstrated that if they took 4 different broad spectrum antibiotics, so imipenem, piperacillin, tazobactam, patients that received the antibiotic were actually developing more graft versus host compared to the patient that received 2 other types of antibiotics, which are also the same broad spectrum, cefepime and aztreonam. And what's interesting also is that they demonstrated that if you receive these antibiotics, you had more mortality secondary to the GVHD, but there was no real change in terms of relapse, further demonstrating that the development of this lethal complication, the GVHD, is microbiome dependent. And then they went further than just saying the diversity is important, but also what type of bacteria are important. And they nicely demonstrated that one bacteria called Blausia was increased in patients, so here that are white, so meaning alive and with no severe GVHD. And interestingly, patients that were lacking this Blausia unfortunately developed a lot of treatment-related mortality, complications that were not related to GVHD, but also in red, a lot of GVHD. So further demonstrating that certain bacteria are key to prevent GVHD. Conversely, another group demonstrated that one bacteria present in the gut called Enterococcus was associated with higher mortality, so shorter overall survival, and more development in a graft versus host disease. Again, in science, what's important is to demonstrate the mechanism. So you have these results in patients, but then doctors, scientists want to know why. And then to validate this observation, they used, again, mice and murine experiments of allogenic stem cell transplantation. And you see that mice that received the Enterococcus, so in red, were unfortunately dying faster than the mice that received the that did not receive the Enterococcus and just the bone marrow transplant. And so once a patient developed graft versus host, mostly, and the most severe is GI graft versus host, so you develop diarrhea. Sometimes it can be bloody diarrhea. There's now new medications such as ruxolitinib, prednisone. And then some people said, but what about giving bacteria? Because we now understand that the development of this toxicity is associated with the gut bacteria. So why don't we salvage and give new bacteria to the patient? So of course, how do you give bacteria to someone? Either you can cultivate the bacteria in a petri dish and then you give it to the patient, but this is difficult, and which bacteria you give. And the only treatment that is currently health approved to give bacteria to someone is for the treatment of C. difficile, so Clostridium difficile. So that's a bacteria that increase after you receive antibiotic. And the way, or one of the strategies to treat C. difficile is to take feces from a healthy volunteer and to transfer, to perform fecal transplant, the feces of the healthy volunteer in the patient with C. difficile. So this group said, but what about doing this to treat GI graft versus host? Because we believe that this is important, bacteria important in this for the development of this disease. And so what they did is that they did in the first patient, so 2 FMT, so they took feces from healthy volunteer, and these patients that had GI GVHD, and you see that just giving feces of someone else was able to stop the diarrhea. And the second patient, again, similar results. They were able to stop the GI GVHD to save the patient life just by giving fecal transplant. And so then there is a French company called Marat Pharma who has developed a drug based on feces where they take pool of 4 to 8 different healthy volunteers and they create a solution that is provided to the patients that have a GI GVHD that is refractory to a steroid. And they presented the first paper that was published 2 years ago, and that they show quite a good response. And last week at the large bone marrow transplant meeting in the States, they presented the second clinical trial. And once again, these are very encouraging data to use the microbiome to decrease GI GVHD. I'm not going to go too much into details, but there has been 2 important papers that also demonstrate that CAR-T cell efficacy and also toxicity, so the cytokine release syndrome, correlated with antibiotic gut microbiome composition. And then I received a question last week by one of the viewer of this talk about what about myeloma and gut microbiome. So I have to admit that we know much less about myeloma and gut microbiome yet, but we, our scientists, are working in this field. And there is one very nice story that demonstrated that patients that were eating bacteria that were not producing short-chain fatty acids. So these bacteria are the bacteria that you need. So when you eat a lot of fiber, you have a lot of bacteria that produce short-chain fatty acids. So that's why, for instance, we know to prevent colon cancer, you need to eat fibers to increase the number of bacteria that produce short-chain fatty acids, such as butyrate, propionate. And this protects the development or prevent the development of colon cancer. And in myeloma, potentially, again, it's probably too early to say that you need to eat fibers to prevent myeloma development, but there was one nice paper demonstrating that patients that had myeloma had less bacteria that were producing short-chain fatty acids. And of course, and this is currently being studied in the field of myeloma, but there is different strategy, and Dr. Elkrief will tell you about different strategy that she's using for solid tumor, but there is similar trial that are opened in Canada, in the States, to try to modulate, to shift your microbiome using different strategy to improve myeloma, leukemia response. And so I will let Dr. Elkrief take over. I'll just, in conclusion, remind you 3 things. So first of all, what you eat is important. Second of all, the good bacteria are important. And a lot of diversity is important. However, what you have to be careful is that, unfortunately, in 2024, we still don't have signature, and you'll see we're working days and nights to understand this, to predict really GVHD development, to predict leukemia development, to predict myeloma development. So I think that besides being part of clinical trial, there's unfortunately no role to get your microbiome sequenced by a private company because we actually don't know what to do with the result. But I highly encourage you to participate in clinical trial at your hospital, to reach out to the nearest academic hospital in order to understand the key role of the microbiome. Because Dr. Elkrief will tell you there are extraordinary results in patients with melanoma.
So now that we've learned about just how important the gut microbiome is in training the immune system, but now the next question is what can we do to actually potentially change a patient's microbiome to improve the responses to cancer treatments? And our lab focuses a lot on immunotherapy, but as Dr. Routy mentioned, a lot of what we're learning can potentially be applied or studied in hematological malignancies as well. And so how do we do this? I think the first important step is looking at biobanking. So what's a biobank? It's a collection of patient samples, and we're really interested in collecting stool samples to be able to learn from patient samples and determine what are the good bacteria, the bad bacteria, and how can we predict the response to immunotherapy. And then we bring these samples to the lab where we test out different strategies and then apply what we learn in the lab to design clinical trials to hopefully help more patients. And this is an example of, for example, our culturomics facility. Culturomics is a study of actually cultivating bacteria. This is important because there's a huge number of bacteria that we know exist, but that have never been named or defined. And so doing culturomics actually allows us to discover new bacteria. These are examples of bacteria that have been discovered as part of Bertrand's lab. And we can see the students that have all been essential in bringing that to a reality. So what is the focus of our work? So we work a lot on strategies to improve response to immunotherapy. And what does immunotherapy do? So essentially, it works on stimulating the patient's own immune system to target cancer cells and to find cancer cells. And now immunotherapy or immune checkpoint inhibitors are used in over 50% of patients with solid tumors, and even now more and more in patients with liquid tumors or hematologic malignancies. And we do a lot of lung cancer work, and there's really been an explosion over the past few years in bringing these cancer immunotherapies not only to patients with advanced disease, but patients with early stage disease, where we can give immunotherapy before the patient's surgery and then potentially make the surgery much easier. And why is this important? So we know Bertrand talked a lot about the negative impact of antibiotics in the context of hematological malignancies, but we learned from that and we're able to apply this in patients with solid tumors. So for example, in patients with lung cancer and patients with melanoma, but here's an example in patients with lung cancer, where we looked at patients that received antibiotics in the 30 days prior to receiving these immunotherapies, and we see that the patients that got antibiotics did worse. So they survived less long than the patients that didn't get antibiotics. And now we're seeing more and more of these combination strategies where we combine immunotherapies with other treatments. So for example, chemotherapy. And this is important because this is really the first study that demonstrated the negative impact of antibiotics, not only for immunotherapy, but also for chemotherapy as well. And what has this changed for us? So it's changed in routine oncology practice that now, prior to starting immunotherapies, doctors are asking about antibiotics. We're making sure that we try to select the shortest course of antibiotics possible, and really make sure that antibiotics are really needed before we give them. And that brings us now to what can we actually do to transform a patient's negative microbiome, one that's associated with cancer, to that of a healthy person's microbiome. And these first experiences started in mice. Bertrand was one of the first scientists to discover this in the lab. And learning from that, we're able to now apply this in patients. So these are two pivotal clinical trials in patients with melanoma that took patients with melanoma that had actually stopped responding to immunotherapy and tried to then re-challenge immunotherapy by giving something called the fecal transplant, which we learned about. And the important point here is that the source of the fecal transplant was actually from patients with melanoma who had a response to immunotherapy. So we're trying to replicate this responder phenotype or this responder status to try to make the immunotherapy work again. And these two studies really provided extraordinary results where we saw responses along the lines of 20 to 30%, which was much higher than what we would expect compared to immunotherapy alone without the fecal transplants. And so we are now replicating this in a slightly different setting. So we have different clinical trials where we're testing this strategy in patients with melanoma and lung cancer, specifically in patients that have never been treated before with immunotherapy. And the other key difference is that our source of fecal transplant is using stool from healthy volunteers. So these are volunteers that bring their stool, donate their stool to our collaborators in London, Ontario, who then formulate these oral capsules of FMT, which are then swallowed by the patient. And importantly, you'll see that all of these capsules are before they're put into capsules, the stool is tested for every single known infection, parasites, bacteria, even COVID and monkeypox. And so this is something that's very important because we want to make sure that the treatment that we're giving is safe. And the design of the clinical trial is as follows. So essentially, a patient that's going to be starting immunotherapy, a week before they receive their immunotherapy, we give something called a bowel preparation. So if any of you have ever undergone a colonoscopy, it's essentially the same bowel preparation, which is a laxative that empties out the colon and the gut to be able to make space for the good bacteria to then take over. And that happens the day before the transplant. And the transplant happens a week before the immunotherapy. And then the patients go on to receive their immunotherapy. And then we assess the response using scans. And this is an example of a real patient. So the first patient that was treated in Montreal, patient with melanoma, who at the time had advanced melanoma. And we'll show the video and then I'll give you a little update on how she's doing today.
Ça a super bien été. Ça m'a surpris. Je pensais que j'aurais de la difficulté, que ça passerait pas. Mais non, ça a très bien été. Je veux dire, et ça allait très bien. En tout cas, je trouve que si d'autres personnes peuvent faire, si on peut apporter des résultats positifs, j'espère que d'autres personnes vont suivre. Je pense que c'est un peu plus d'espoir que ça donne à notre vie. Ça a super.
So as you can see from the video, these are oral capsules. There's no, gross factor. These capsules are triple encapsulated. It means that they're encircled 3 times, which means that the capsules don't have any smell, any taste, and they're designed to release at the beginning of the small intestine. And so that really makes sure that it's safe and it's very well tolerated by all of our patients. And so these are results from the first study in patients with melanoma. And this is what we call a waterfall plot, where each of the bars represents a patient, a real patient that participated in the study. And what we can see is that in blue, there's 4 patients that had what we call a complete response, which means that we're unable to detect any evidence of cancer on the scans. And when we look at the overall response rates, we're seeing a response rate of 65%, which means that 65% of patients benefited from this treatment. And when we compare to our benchmark clinical trials, we're seeing improvements of about 20%, which are very encouraging. And importantly, we see that the combination of the fecal transplant with immunotherapy was safe. And so building on these results, we've launched a newer clinical trial, which has actually now recently been complete. So we've now enrolled all patients. We have 3 different groups. We have patients with lung cancer, patients with melanoma, but this time treated with a different regimen of immunotherapy, and patients with eye melanoma with the same design. And we're seeing, again, very encouraging results, and we're going to be testing this now in a large study of patients with melanoma across Canada. And, as an oncologist in the clinic, patients read, about these studies, and there's a lot of interest now in the microbiome. And one of the questions that we often get asked is, what are the things that I can do if I'm not participating in a clinical study? We go to the health food store and we see all these supplements. The truth is that there's really never been any studies that have tested these different supplements as part of randomized clinical trials. And so there's definitely no recommendation to start taking these natural products. And I also wanted to clarify a little bit, what are the differences between probiotics and prebiotics? And this is important because we hear a lot about these different products in the news, and it's important to know what's what. So probiotics are actually actual bacteria that can either be present in the form of capsules or they can be present in the form of different food products. And then there's prebiotics, which essentially is any compound which can beneficially shift or change a patient's microbiome. And this can be in the form of food, but it can also be in the form of natural supplements. And I think what's really important to know is that none of these products are really approved by Health Canada. They're not regulated. We really don't have firm guidelines on how to regulate these products. We know that there are multiple issues with potential contamination and safety issues. And so even though I think the key message here is that even though these products can be found at health food stores and we can just walk in and buy them, it doesn't necessarily mean that it's the right thing. And there could even be some potential safety concerns with regards to these products. And, this is an example where we actually looked retrospectively at patients that received probiotics that were treated with immunotherapy, and the patients that took probiotics actually did worse. And so important to keep in mind, please talk to your doctor and your pharmacist if you're taking any supplements, because they could interact with different treatments that you might be taking for cancer. And so we're really, really passionate about how we can use clinical trials to change patients' microbiome. This is true for lung cancer, melanoma, and currently being studied in other contexts as well. And we have fecal transplant, which we spoke about, probiotics, which are actually testing different specific bacteria that have been associated with response, prebiotics, looking at different metabolites, and of course also how can we potentially change a person's diet by working with a nutritionist to try to increase fiber, for example. So these are examples of some of the projects that we're working on. And so to conclude, we know that now the field has really exploded with data and evidence linking the microbiome to outcomes for solid cancers, for liquid cancers, and that these beneficial versus deleterious bacteria are really important in predicting response to treatment. We know that we now need to learn more and more from patient samples to be able to answer these questions. We need to be really judicious with antibiotics. We need to choose the right antibiotics for the shortest amount of time because we know that antibiotics are harmful. There are many different strategies and ways to potentially improve the patient's microbiome, to improve the response to immunotherapy and different hematological interventions, and potentially also toxicity like graft versus host disease. And besides general health recommendations like eating healthy and exercising, to date, there really haven't been any proven interventions to improve the microbiome. And so that's why we're working on all these clinical trials. And with that, we'd like to, thank the team, all of the team that helps us in the lab and the clinical trials, and our collaborators, and of course our funding sources. And thank you so much for your attention today. It was a pleasure to speak to everybody.
Thank you both for sharing your expertise and insight. This is certainly a very complicated and complex topic, and we have many questions have come in this afternoon. So it is time for our Q&A period. If you haven't done so already, please be sure to use the Q&A box to put your questions in. We'll be monitoring them as we're going here. As we noted before, no personal questions will be answered, and any questions that are not related to the topic will be answered offline or taken out of the list. So we've had lots of different questions, and I'm sure you can imagine where they're all coming from. But we'll start with maybe general and get more specific as we're going through here. And to start, so you mentioned that probiotics, prebiotics, supplements are not impactful for this work. Sorry, but if we're looking at the best overall diet, I guess, plan, if we're keto, intermittent fasting, anything like that, those words get thrown around a lot. There's certain specific foods that are more beneficial to than others. So I don't know if you can talk to that generalization of diet and the impact that this will have on your gut microbiome.
Yeah, so the problem is that what we have to recommend is based on scientific and randomized trial, and not on the desire to become rich. And unfortunately, in this field, a lot of people are trying to sell to patients, and some of my patients receive email by special leaves that is amazing, very expensive tea that monkey are picking up in New Zealand, and they ship you this tea by Super FedEx, and it costs you $2,000, and you're going to be cured. So unfortunately, there's a lot of fake news out there regarding the diet or amazing product. So that's the first fact. The second thing is, what about the diet? And it's quite difficult, because even if I tell you Mediterranean diet, it means something to you, but what's the real Mediterranean diet? Do you have to live in the south of France like I'm for to have a Mediterranean diet, or it's possible to have a Mediterranean diet in Canada? Question mark. So we don't have the definition of these diets, so that's very important. The second point is that these diets are currently being studied in clinical trials. We're just having a meeting with the group from MD Anderson, who are doing a high fiber diet trial in the States. There is other trials that are ongoing. Unfortunately, and I'm very sad to tell you that I receive a lot of requests from a publishing company and books. Oh, Dr. Routy, please, let's write together a book on what to eat before your immunotherapy, what to eat before your bone marrow transplant. And I tell the editors, I'm sorry, but the book is not going to be very exciting because I'm going to say, I don't know. And besides following the Canadian Nutrition Guideline, that's the only thing I can tell the readers. And the ASCO guidelines, so the American Society of Clinical Oncology, actually wrote black and white, there is currently no indication to do anything to improve your diet. And there's no strong recommendation to do anything. So that's why we are very cautious. The fasting, you know, when you get a bone marrow transplant, I'm not sure it's the best moment to fast because then you get into kidney failure. The ketogenic diet, again, what's a real ketogenic diet? But this has to be tested in randomized trial. You have to be part of a trial, I think, to do this type of diet. And I don't recommend anything to my patients besides participating in my diet. And maybe, or at least to follow the Canadian nutritional guideline. That is the best thing we can recommend patients. And of course, don't smoke and remain active.
Right. So you recommend a variable, diverse diet, including all kinds of different foods and to help, as recommended. Very good. So I guess you touched upon this, and is the clinical trial space, and how do people find clinical trials if they want to be involved with this work in Canada and abroad, especially, I suppose.
Like that.
The first way, and probably the most straightforward way, is to talk to your doctor. Number one, talk to your doctor to see if you would be a candidate, if you would meet the different criteria, if they're aware of any clinical trials in their network. The second thing is that there's a website called the Clinical Gov Network, which is basically a database, a national, international database of all the clinical trials. And that can be filtered based on location, based on disease type. We can put it in the chat, and that can be shared. And that's a free, publicly available database that patients can search and then go to their doctors and ask if there would be an indication to participate in such a clinical trial. And then the other aspect is, just using a search engine like Google. There are different patient advocacy networks that have a list of clinical trials depending on the different disease sites. So for example, lung cancer has their own, Lung Cancer Research Canada has their own, and I'm sure that there's an equivalent for different hematological malignancies within Canada.
Thank you very much. Okay, so again, we have lots of questions about treatment here, and we'll start with chemotherapy and the impact that actual chemotherapy has on your gut microbiome, and how we can help recover after chemo. What is the best way to approach this?
Once again, so the reality, and I'm sorry for the audience, but we don't know, and they're going to say they know nothing, these people. But so we don't know. We know that chemotherapy decreases diversity, and I showed you the graph. Then what to do after when you come out of neutropenia, when there's the engraftment, it's still unknown. So the French trial, the MAD trial, they're doing FMT, so they take your feces and they give it back to you after you're coming out of neutropenia. The only thing that we know to not do is to take off-the-shelf probiotic. I think that the study from the Americans, from MD Anderson in melanoma, was to me a red flag to really, and I've never recommended any probiotic, but this was to show to the patients, to my patient, that if you go and buy yourself at the drugstore a probiotic, it actually could harm you. And so that's why, again, I do not recommend probiotic. I do not recommend prebiotic, because currently, and unfortunately, even for patients that take antibiotic, there's a lot of companies that want to sell you these beautiful probiotic that you can buy at any drugstore, and they tell you, oh, you'll recover much more your microbiome. But we know there was a nice paper from Israel where they demonstrated that patients that receive antibiotic, those that took probiotic took more time to recover to their baseline microbiome compared to those who did nothing. So unfortunately, in 2024, if you're not part of a clinical trial, there's no recommendation to take probiotic when you're on chemotherapy or when you're on any cancer treatment, because there's no real proof, and the only evidence we have is that it could be harmful for you.
And so does that apply to supplements like vitamin C and omegas and all that range of things as well for chemotherapies?
So that's another thing. And we have not talked about supplements. Again, if, first of all, you have to tell your doctor, but if you have a normal diet, in theory, you would not require any supplement. And in Canada, the quality of the food is good enough that if you drink and eat fruits, you have the vitamin C. If you eat meat or beans and lentils, you have the iron. So in theory, nobody should be on supplement pills if they eat adequately. Then also a possibility, when you have cancer, you don't eat as well. So then you go see a nutritionist, and she might recommend you a specific supplement. But again, this is not for your cancer treatment, and this supplement is not for the microbiome.
And so in relation to the microbiome and the connection to our bone marrow, and we're talking about hematological malignancies here. What is the connection between a healthy gut microbiome or an unhealthy microbiome and bone cancer, or bone marrow, sorry?
So that's a very good and challenging question. So we think that the immune system is educated. So if you remember from Physiology 101, but your T cells are going in your thymus, and those that recognize yourself are destroyed, and those that do not recognize yourself are allowed to go in the circulation. And now there's new evidence that these baby T cells can go along your GI tract, and they will get educated with the bacteria they see. So definitely your immune reaction to anything, to allergy, to potentially development of cancer, is probably dependent on your gut microbiome. And then what is still unknown is what about the development of cancer? So there's a lot of papers, especially in pediatric leukemia, where they look at the microbiome of the kid that has the leukemia, and they compare it to the microbiome of the leukemia-free brothers or sisters, and they have a different microbiome. And of course, it's the chicken or the egg, and we don't know it yet. Our team is actively working on this. We're now aiming and writing a grant to collect microbiome of everybody who's at the age of 50, and then to follow this patient over time and to see who's going to develop microbiome. And hopefully, you will invite us in 10 years, and we're going to say, at 50, we knew you were going to have leukemia because you have these bacteria. So now, if in 10 years this program works, we will say, okay, at 50 years old, instead of getting a colonoscopy, a rectal exam, a mammogram, we will just look at your microbiome, and then, bacteria A and B are increased, or you're at very high risk of developing leukemia. Maybe let's do blood tests every 6 months. Bacteria C and D are increased, or your risk of having a myeloma or MGUS are increased. So make sure you have a protein electrophoresis. So all these types of preventing the development of cancer through microbiome is my goal, but again, too early to comment on this.
Right. And I assume this will all play into GVHD and whether it has an impact on remission or duration of remission.
So again, most data are demonstrating the impact of microbiome for GVHD, not the impact of the microbiome on graft versus leukemia, which is key for remission. So this, what we still don't understand, is why the observation from the antibiotic papers or from the microbiome sequencing, we don't know which patient is going to relapse or not based on their microbiome in the allogenic stem cell transplant. We just can predict potentially those that will develop GVHD. In solid tumor, so Dr. Elkrief is putting a program to try to, the new words in the street is liquid biopsy, so to try to predict development of cancer beforehand by looking at the blood cells and see if you see cancer cells. But what about the microbiome? Could it be like the second generation, the future generation of liquid biopsy or semi-liquid biopsy? And to, prospectively profile the microbiome of patients that are undergoing allogenic stem cell transplantation. And you can ideally, or that's what they're doing in the States in certain centers, to aim to predict early relapse based on your microbiome composition.
Thank you. And so I think we have time for one last question here, and let's end this with a message of where things are going in the next. You touched upon this, but in the immediate future, 5-10 years from now, where do you predict this field will take us?
So the field is still in infancy, but in medicine, this field has moved very fast. So I can tell you that 10 years ago, when I left Princess Margaret, my boss told me that I was going to destroy my career to go in France to work on the bacteria. That was stupid and I should not go there. So that was 10 years ago. Now all the big pharma are really interested in the microbiome. Dr. Elkrief just joined us from New York to work on the microbiome. At every major symposium and international conference, there is a half day or session dedicated of the microbiome. And clearly, to us at least, it's really the second paradigm shift in immuno-oncology. And I think that once we'll be able to understand the mechanism, companies will come, new device, new strategies to really have a personalized microbiome treatment is the future. And I can tell you that in the next 5 years, there will be a lot of microbiome-derived drug in cancer. And hopefully in 10 years, at the time of the diagnosis, they will tell you, okay, a blood sample, a bone marrow biopsy, and a fecal samples to able to, predict, do you have good marker, bad marker? Do you need bone marrow transplant upfront? Are you going to respond to it through the integration of the microbiome? With, of course, every other markers that are currently available.
Okay, well, I think we'll end it with that as a message of hope, and this is certainly a fascinatingly complex subject, and we'll hear lots to come from this area in the coming years. But thank you once again to both of you for being with us here today. And I'd like to remind everyone that we are here for you. Do not hesitate to contact us if you need any more information or support concerning this topic or any of our services. Please note that a short survey will be sent to all participants after this webcast. We would greatly appreciate you taking a moment to fill it in, as it will help us to offer you with the most valuable information in the near future. I hope you all have a wonderful rest of your day. Thank you very much.